Okay, welcome back. We're going to talk about the genetics of quantitative characters and how we can interpret that in evolutionary terms. First of all, what do we mean by quantitative characters? Most of the really important characteristics, phenotypes that are relevant to medical sciences are quantitative. We've been speaking so far about qualitative characteristics in which either one thing or the other thing is the phenotype, but many others can be measured on a continuous scale. Here I'm showing some data collected in the 1940s from University College Hospital in London by Mary Kahn on the birth weights of babies at University College Hospital over that decade. And what you see is that the birth weights vary over a very wide range. I'm afraid this is in pounds weight rather than kilograms, because that's the units they used to use. But you can see that there's a distribution, a sort of bell-shaped distribution, with an average baby weight where most of the babies are that sort of weight, and they become increasingly rare as they get to extreme weights. And that's something we observe for many human characteristics. They went one step further, which was a very important step, and asked the question, what's the survival rate of babies of these different weights? And that's shown in a green curve on this diagram. And what you see there is the survival rate of babies of roughly average weight is really very good in the 1940s. But as you get to babies that are really rather light, rather small, the survival rate drops away quite dramatically. Of course, those rates would be different now, that the survival rates would be higher with improved medicine, but nevertheless a similar sort of relationship would hold. And you can see why. Either the very small babies had many things wrong with them, or they were born prematurely, or something went wrong during development and so on, and so they have a lower survival rate. What they also show, as you can see on the diagram, is slightly odd and interesting, is that survival rates begin to decline with the more extreme heavy babies as well. Why might that be? Well, apart from there could be conditions which lead to high baby weights which are deleterious, also giving birth to large babies is a more risky process, and so you can see why there would be some dangers associated with excessive size of babies, which of course you could do something about also by improving procedures. So what you see there is there seems to be a disadvantage to being a very small baby or being a very large baby. Now, to the extent to which the size of the baby is determined by the genotype of the parents, then you can see that evolutionary selection pressures could act to maintain a genotype distribution which gives you the phenotypic distribution you see here. So the conception is that the mean value of the observed of the phenotype in the population is somehow optimal. It's sort of a Goldilocks value. It's neither too small nor too large. It's just right for survival, and that selection pressures are eliminating those genotypes which give rise to extreme values of birth weight on either side, and tending to push the distribution generation by generation towards a mean value. This is something we call balancing selection, and it's a very important property in quantitative genetics. However, when they looked in more detail statistically at those survival rates, they determined that the optimal weight for optimal survival was actually 8 pounds rather than 7 pounds. This figure on the right here comes directly from their paper. So that's a higher weight than the mean weight of the population. And the question then begins to occur to you, why is the optimal weight not the average weight of the population? It could be, for example, that changes to medical practice over the years mean that it's now all the disadvantages of being a large baby have gone away, and so all the advantages of being a larger baby outweigh them, and compared with the past, and so the optimal weight is gradually shifting to the right to higher weight babies. It could be that, or it could be something like the larger babies, although they survive birth better, don't survive for one reason or another. They don't reproduce as well later in life. That's a possibility that would need to be explored. So it's considerations like this that one has to bear in mind when looking at quantitative characters in populations. So having looked at one particular quantitative character, we can generalize. How do we describe and analyze these populations? As you saw, they are distributed frequently according to a sort of normal or Gaussian distribution, and normal distributions you will have heard of can be described by two major parameters. One is the mean point, the middle point of the distribution, and the other is a measure of the dispersion or the variation in the population. And the standard measure is variance, which is symbolized there as sigma squared. And the square root of the variance can be plotted on the same graph as the mean is plotted, and it's shown there as sigma on that distribution. And in fact, we very often use units of standard deviation as standardized units of measure on continuously varying characters. We've spoken about characteristics whose phenotypes can be measured and can be placed on these sorts of distribution curves. We can imagine that some characteristics can't be directly measured, can't be observed directly, but somehow underpin other characteristics which express at some stage during people's lifetimes. So, for example, we could imagine that there may be underpinning quantitatively varying characteristics such as risk factors for disease, which vary in the same sort of normal way as observable characteristics, but are expressed in a qualitative form by higher probabilities of developing the disease at one end of the spectrum and lower probabilities of developing disease at the other end of the spectrum. How can we analyze, how can we model the genetics that might underpin this sort of variation, to the extent to which genetics does underpin any of these characteristics? Well, a model that goes back to R.A. Fisher is as follows. We could imagine a case where we had just one gene, like the gene A you saw before, but in this case, instead of it having two alleles which are either or alleles, the two alleles in this case are increasing or decreasing alleles for a quantitative characteristic. I'm showing you here the two alleles 0 and 1, and when you have an individual that carries two 0 characteristics, they have a phenotype of 0, and when you have an individual with two alleles 1, they have a phenotype of 2. And individuals that are heterozygous have a phenotype of 1 because there's incomplete dominance between these two quantitative characteristics. And if the two alleles were present at equal frequency, your population distribution would look like this diagram, with twice as many people of the intermediate phenotype as those of the two more extreme phenotypes. And I suppose environmental variation might affect the phenotype so that those three peaks would smear out a little bit, but it still wouldn't look like a normal distribution. However, if you increase the number of genes that are governing this quantitative phenotype, you find that you can approach the normal distribution quite quickly. By the time you've got to 5 different genes or 5 different loci, each of which has a pair of alleles, an increasing allele and a decreasing allele, you can simulate a normal distribution really very easily, as shown here. So this sort of theoretical analysis, sort of assumption, has been at the basis of much of quantitative genetics over the years, and enables us to analyze and model certain key properties of populations and the genotypes that underpin their quantitative characteristics. And one of the most important properties is known as heritability, the extent to which the characteristic in question is governed by the genes of the individuals carrying that character, expressing that characteristic. So characteristics that have a very high level of heritability will be very largely determined by their genes. Characteristics with very low level of heritability would be largely determined by the environment that the individuals find themselves in. How can we understand, work out, analyze and determine heritability? Well, first of all, let's begin to define our terms. You remember I spoke about normal distributions being characterized by a mean and a variance. Well, the variance of characteristics is known as the phenotypic variance of the population, VP. And we can think of that variance being made up of a genetic component and an environmental component. If we take a concrete example of varying phenotypes, here's one from John Hawkes' weblog where he's been measuring some of his anthropology students. Each of his students is shown here as an orange dot, where their position on the y-axis is determined by their height as measured in class, and their position on the x-axis is determined by the height, the average height of their two parents. You see there's quite a scatter. There's a scatter in the y dimension, which is a sort of normal distribution of heights of the student population, and there's a scatter in the x dimension, which is a normal distribution of the heights in the parents' population. But when the two are plotted against each other, you can see, although the scatterer is quite broad, there's a tendency for the two heights to be correlated. That is, there's a tendency for tall parents to have tall children and for short parents to have short children. But it's not a perfect correlation. And you'll know that you can look at these sorts of relationships in terms of regression analysis. So if we do a least squared linear regression of the offspring height, the student's height, against the average of their parents' height, the slope of that line is, in this case, 0.72. And that's an interesting feature. It seems to relate to how inheritable the phenotype of height is in these families. So if we go back to the left-hand side of the screen now, think about the variance of the phenotype in the population being composed of genetic variance and environmental variance. In other words, there's a sum of these two factors. We just need to dissect the genetic variance one more bit. Genetic variance we can consider being made up of two major components. There's some others that can come into more complex realistic models, but if we think of the additive genetic variance and the dominance genetic variance, VA and VD, additive genetic variance is the genetic variance that is conferred simply by the phenotype of the alleles in the homozygous cases where the heterozygotes have an intermediate phenotype, and the dominance genetic variance is the extent to which the phenotype of the heterozygotes is not intermediate between the homozygotes. The heritability is often shown as the expression h squared, which is like sigma squared. It's a variance. It's a variance ratio between the variance due to the additive genetic variation in the population divided by the total variance of the phenotype in the population. That's what you'd like to get, and it turns out that it's the same as, it's equal to, given some assumptions we'll come on to in a minute, it's equal to that regression coefficient of the value of offspring characteristic to the mid-parent characteristic that we showed in a diagram like that one. So narrow sense heritability, the thing that responds to selection pressures, for example birth weight ones, is the ratio of the additive genetic variance to the total phenotypic variance, and it can be estimated by doing a regression of offspring values against mid-parent values. But there are some very significant caveats which we'll come to in a minute. This sort of study, offspring versus parent values, can be done for a whole range of human characteristics of interest to medicine. Here I'm giving you just one example, published in 2002, of neurocognitive functions in a population in northern Finland. This was chosen because it was a nice isolated population which had some interesting high levels of schizoaffective disorders. It was a very well studied population. They were able to establish pedigrees going back several generations for all of the people in the study, and they analysed all of the members of this population psychometrically and did the regression analysis we spoke about in the last slide of the values established for one generation against the mid-parent values of the previous generation. And they developed a table like this one. Let's just look at that. There's quite a few different psychometric tests shown here. At the top there we've got one recall errors, and that one, if you look at the third column, has a high heritability level. So 0.66 heritability. What that means is 2/3 of the phenotypic variance in this study population for that variable, recall errors, could be ascribed to additive genetic variance in that population. It's very high level heritability. Whereas down at the bottom of the table, visual attention, for example, the contribution of additive genetic variance to phenotypic variance was extremely low. There was very little evidence for any genetic effect in this population for that particular property. So this can be very interesting. We can ask questions about what are the environments now that affect things in the lower part of the table, and we can ask questions about what are the genes affecting things that exist in the upper part of that table, and maybe identifying these genes can identify therapeutic approaches to intervening in the development of those diseases. But as I said before, there are some very strong caveats. These sorts of studies are very imperfect and are based on some assumptions that may not be safe. So first of all, they assume that there's no covariation between the environments and the genotypes. And we know that isn't entirely true. We know that families generate their own environments, so that children share environments with their parents that they don't share with the children of other parents. And so the covariation of environments and genes in study populations can result in an overly high estimation of heritability. And this is a well-known factor that has to be taken into account in studies. Secondly, and this is really critical, many people are inclined to think that if a character or a disease has a very high level of heritability, it's somehow or other an inevitable characteristic. Oh, we can't do anything about it, it's all in our genes. That is not a safe conclusion. All it says is that the heritability of this character in this study population, with its variation in genetic composition and its environmental variation, is highly heritable. If we were to change either the genotype frequencies or the environments, we would change that value of heritability. And it's the change in environments which is really important. It's, after all, what medicine does. It changes the environments in important and targeted ways. For example, bringing in something like insulin for the treatment of diabetes. Before the innovation of insulin, you were entirely dependent on whether your genes and the interaction of them with the environment led to type 1 diabetes. Once insulin comes along, we can manage the disease relatively well. So it changes the phenotypic expression of the genes in the population and changes the heritability. So just because something's heritable doesn't mean to say there's nothing medicine can do about it. But secondly, you have to bear in mind that the heritability is a property not only of the environments under study, but also of the genes under study. And in that study we were just looking at, we were looking at a very convenient population in northern Finland, which can be studied. But it could easily be that the genotype pool in that population is very different from the genotype pool in other populations. And in other genotype pools, the heritability might be very different. So we have to bear in mind that the heritability values we look at from one study may not apply in all populations. So, I hope I've given you a very brief introduction to analysing quantitative characteristics and an indication of how we can use some of these sorts of studies to try and identify either environments or genes which contribute to disease characteristics, and this can lead on to developments in medicine.